For the DNA transcription and translation assignment, you're going to start by grabbing the paper that you see in front of you. And then you're going to need to cut out each of the shapes that are on that paper like you've seen here. And then finally, you cut the strands of DNA apart and connect them 1A to 1B, 1B to 1B, etc. Next, you're going to grab the template strand that has the mRNA and the protein chain, and you're going to cut one of those three objects out. Notice that I kept the gray attachments so that you can cut those apart and tape the mRNA strand together. And then finally, you're going to start the process of mRNA transcription, which you will see a video of in just a second. To get this set up for the transcription phase, where we copy the template strand of DNA into a strand of mRNA, you are going to take the DNA strand and you're going to run it through the slit that you made so that the top of this RNA polymerase molecule is blocking the top strand of the DNA, and all you can see is the bottom strand of the DNA, and you're going to line it up so that you can only, so you start with the circled letter. So whatever your letter is that's circled in green should be the first letter that you see. And then laying on top of that, we're going to set our mRNA strand that's unlabeled. And you'll have it laid on top, and then grab a piece of tape and tape the left-hand side together. So don't tape it to the polymerase, but just tape that little strand of the mRNA to the template strand of DNA, and then you'll be able to kind of pull this RNA polymerase along to sequence your genome, and you'll just be marking out each corresponding pair on your template strand. Once you've finished transcribing that mRNA molecule, you should see the whole thing written out as a sequence like you see in the picture. And you're just going to separate those two strands again. So now you're only looking at the mRNA strand, and you're going to find that start codon, which is the AUG codon. So you need to find that and circle it. And then you'll start circling every three-letter sequence because those will represent your codons. And once you have your codons, you can use that mRNA, that ribosome, and you can cut out the window, and you'll be able to sequence one codon at a time. And you'll use your codon amino acid wheel to figure out what the single letter code is of each codon for your amino acid. And you'll write that down on your amino acid chain sequence. And then you will tape the amino acid chain sequence into your notebook along with the other objects as well. Thanks for watching.